And maybe it's your smile. Is there some trait that is not simply mom's or dad's, but looks a little bit in between? Maybe it's your skin color, right? Does anyone have a situation where one parent might be darker or lighter than the other, and you are in between the two? Right, and what of your eye color? Is your eye color a perfect blend of both parents, or a mix of those two things? Yes. Thank you. It's a very important message. Peacekeeping in the Middle East, right? Making sure you know the house and I don't really joke about. All right, so, and in thinking of those traits, we're going to examine that today. So in middle school, you had learned about dominant recessive. We've just been reviewing that. Now we're moving on to the more high school topics, the ones you'll be assessed on for the MCAS. In terms of our code year, everyone has the obligation to ponder his or her specific traits of character. This is the point of our check-in every day. You are now at the age where you can no longer blame mom and dad for your problems, right? You spend very little time with them during the day. You're here all day, then you're off doing stuff with your friends. Some of you have internships you've started, right? You can't blame your success or failure on them anymore because you are an independent individual. You have to look at your attendance. You have to look at your attitude. You have to look at your productivity. And then you'll notice here we've got two different homework assignments, one that everyone will do, and you'll have class time to start that. The last two periods today, most people finished about 3/4 of it before they left. And then my honors folks, because they are high and mighty, have an additional little thing to do. But based on today's lesson, they'll be able to skillfully master that in a snap. Timeliness and manner. So now we've had some time to think about this. Does anyone have a trait they can think of that doesn't look perfectly like either parent? Something different, something unique to you. Have you ever even looked at those people? Do you even know what they look like at all? You just basically take their food and money and shelter and ignore them otherwise.
What was your question?
A trait that doesn't look like either of your parents, like something unique to you as an individual.
All the computers are there.
Because no one ever plugs them back in. Yes? I've noticed everyone just leaves them on the table and expects them to come back tomorrow and be magically full again. All right, so clearly folks need a little more time to think about that. So we'll revisit that later on. All right, so let's talk about some traits. For now, you'll notice there's some colors on the screen. Don't worry about that at the moment. We'll bring that in when we do our drawing later on. So first up, we're going to talk about co-dominance. When you see the prefix co before something, what does that mean? If you are collaborating, cooperating. Or like the Spanish conjunction con. With, right? So you are with something. So in this we are saying with dominance, right? So this is, again, a different situation than we've been learning about in that rather than one allele being dominant, taking over, and one allele being recessive, being covered over, both share the stage equally. For example, if I have two colored pencils, a blue and a yellow, and I put them both on the paper on top of each other, is the paper only blue or only yellow? No, it becomes yellow, blue, green. Very nice, right? And that's a good example of co-dominance. Neither overpowers the other. They share the stage equally. And this happens in traits all the time. For example, in our species, if someone in curly hair and someone with straight hair mate, the offspring's hair is what? Often wavy, right? It doesn't come up as curly or straight. It has a blend of the two. And let's use hair as an example. right? So we think of traits as normally being dominant or recessive. What would you call that if we've got like a mix of the two things? If you have both the dominant allele and the recessive allele, we call that a heterozygote. Now in codominance, what we're saying is rather than big H, little h, it's big H and Q, for example, right? Both these different traits have equal power. They are both capital. And again, this Q, is there like a reason I chose Q? You guys made some of your own Punnett squares last week. When you're representing alleles, you can do any letter you want, right? Like I could put Z or T or L, right? So remember, these are just convenient tools, not the way it has to be. Now you can also be what's known as incomplete dominant. What is it when something is incomplete? Let's say you hand a homework in that's incomplete. It's not done, it's not finished, it's not full, right? It's not whole. So in incomplete dominance, neither takes over fully, and just like this one, they're both sharing the stage. So things are starting to get a little confusing now, because we're saying that in both situations, both alleles are being expressed at the same time. Neither takes over the other. So let's see if we can master this situation. So this is a form of inheritance where heterozygotes are expressed as a combination. Right, so what you're seeing is not a mix of the two, but the two are both together. So, for example, again, if codominance, we have a blue and yellow equaling a flower that is green, right? So we talked about that one already. In this situation, our blue parent flower and our yellow parent flower, rather than producing a flower that has all green petals, what would this one make? So we've got a bunch of petals on our flower. Some of them are going to be blue, and the others will be yellow, right? So, so you'll have a situation where this might be either blue or yellow for our flower. So let's, of course, make a visual for this. On your table, you will notice at your station there are 3 coloring tools, 2 primaries and a secondary. So on your table, you might have blue and red, which makes, or yellow and red, which makes,
What if you have none of the above?
then something bizarre happened because in the last period that was what was distributed. All right, so,
Wait,
what?
I'm not.
All right, I'll go back for a minute. Wait, you've got a computer right in front of you. Have one all right, so here we have a flower that is both red and white petals. Which one would this be? Would this be our incomplete or our co?
Incomplete.
This would be our incomplete, right? And then here we have, whoops, whoa, and then here we have a codominance where the red and the white made pink. All right, cool. So let's draw ourselves some scenarios in order to make this happen. So I'm going to make a little divider here. We have 2 pictures side by side. One picture is going to, I'm going to put this right under your notes. Right, one picture is going to be codominance. Oh, good. Smartboard is missing line, my favorite. All right.
Just go see.
CV. Well, but the thing is, I'm going to be drawing, so I need the thing to be accurate, or as accurate as I can. All right, so we'll put codominance on this side.
Mr. Papaga,
Yes.
may I sit next to Jasmine so I can take down the notes on the computer?
Come on up. And incomplete dominance.
You say yes, John.
So, all right, or incomplete dominance. Awesome. Okay. Now, again, if you have 2 primaries and 2 secondaries, you can create hybrids. You can create mixes. So in this square for codominance, we're going to draw 3 flowers. Our first flower is going to have this symbol in it. So what does that indicate about it? It's a male, and male flowers release what as their gamete? Pollen. Awesome. All right, and then over on the other side of the box, I will draw a female flower of the same species, because again, you have to be the same species to mate. And just like females in our species, female flowers have eggs. They have, ooh, awesome. All right. All right, now let's say I'm going to pick a primary color. You'll do the same. So either you can choose red, or you can choose blue, or you can choose yellow. I'm going to choose red because that's convenient for my smart board. And I'm going to color this male flower in red. All right, so I'm going to say that my male flower is red in this scenario. Now my female flower, I'm going to pick a second primary color. Again, you can use your blue, or you can use your yellow, or you can use your red. I'm going to use yellow. And I'm going to make her this color. Right, so I have one red male flower and one female yellow flower. Now let's say some pollen from this male flower found its way onto the pistil of the female flower. What would happen? You'd make some offspring, right? Very nice. So these guys will make this little flower. It's their baby. Now, in a co-dominant situation where the two genes blend together, they make a hybrid, what color would my baby be? Orange. Very nice. Right, so if you've chosen red and blue as your parents, your baby would be? Purple. And if you've chosen yellow and blue, it would be? Green. Very nice. Right, so on your paper, so we'll all wind up with something slightly individual, right, today, where you've got two primary colors as your parents and one secondary color as your offspring. So let's take a moment and make that happen. I'll come around and give you dirty looks if you haven't done it yet. And if you are injured or ill, you're welcome to go to the nurse or guidance. But of course, your teacher is working together to allow students to have a nice nap in his class. We're all miserable and suffering. But the main purpose of school is to teach you how to sit for 8 hours and do what you're told by a boss. Wait until you start feeling familiar. I bet everyone else can do that. Everybody can do it. All right, so you should have a primary on either side and a secondary in the middle as your offspring. How do I know who are the parents and who's the child? Perfect, right? So one of your classmates came up with a good idea, right? They're also adding in some labels, right? You can make this parent 1.
Take one Beckles, please report to the main office.
All right, I saw one of your classmates do something very clever. I'm going to do the same. I'm going to model after them. Right, so they've written parent 1 and parent 2 equals offspring. All right. Now you can use the same two colors you did before, or different ones if you like. But let's create a similar scenario for our incomplete dominance. So once again, I'm going to have a parent flower here, and I'll make him a male. What do you guys remember from flower reproduction? Are flowers strictly male or female? No, right? They can have both, right? They'll both have both pollen and eggs. But in this situation, we're calling this the male because that's the one releasing the pollen. All right, and then we'll have a mate for him. We'll have a female. So we have once again parent 1 and parent 2. All right, I'm going to use the same colors I did before. But you, of course, are welcome to choose different primary colors for this scenario. Do what works best for your learning. All right, so I'm going to say that parent 1 is red. And parent 2 this really muddy looking yellow. That is remarkably hard to color the lines on this one. right? So again, you're choosing primary colors for your parents. So you might do blue is one, red is the other, or blue is one and yellow is the other, or red and yellow. Now, unlike our first scenario, where our offspring was a perfect blend of the two, they became a hybrid, they mixed. What will my offspring look like in this situation? Some will be red and some will be yellow. Great. Right, so let's go ahead and do that, right? So showing that the offspring shows a little bit of each parent. And the way this usually pops up on the MCAS is they'll ask you about a scenario where two parents have mated. It might be, rabbits or flowers or wildebeest. And they'll say, the two parents have mixed and the child is a perfect blend of the two, looking like something new to the scene, right? And that would be, if they're talking about something new, it's codominance, right? We have a new color here. If they're saying that it exhibits things where it looks like both parents, we have our incomplete, right? It's got red petals and yellow petals. All right, nice. Yeah, and I'm seeing some different combinations from your classmates. So some people are doing like half of them red on this side, half them yellow. That's also acceptable. You could have also done it where you had like part of it be red and then part of it be yellow. So it was like, yellow in the middle and red on the outside. Either would be acceptable as a way of drawing incomplete dominance. Nice. All right. Oh yeah, we have plenty more. All right, so next up we have a condition known as polygenic. Folks already know what this means. Poly, of course, means, from math, we have mono and poly. Or, keep going, right? So mono meaning single and poly meaning plural, or right, many or two or more, right? So we have poly, and then of course genic, meaning those little things, genes, right? So we're saying many genes. So polygenics are traits that it takes more than one gene to make happen. It's controlled by multiple alleles. So there are a lot of things about us. It's not just simply you have a single gene that makes this thing happen in your body. For example, last week we talked about thumb shape. What are the two thumb shapes a lot of us have?
Curve and straight.
You either have the straight or the curve in hitchhiker's thumb. If you have a hitchhiker's thumb, that's not simply a single gene, right? You need one gene that's going to control for the shape of the bones in the fingers. What were the finger bones called again? I remember from back in chapter 2. Phalanges, right? So you have one gene that's going to control the shape of your phalanges. A second gene that's going to dictate the shape of this amount of space in the joint. Another gene that will control how tight the tendons are, right? So how much you can pull on those bones, or I'm sorry, ligaments. How much you can pull those bones, right? So many traits have multiple things involved. Blood type is another example. Can folks remind me, what are the blood types? There was A, what else? There was B, and is someone else? O. Is that the only part of the story for blood types? Is there just A, B, and O? Wasn't there something else? You also have to consider whether or not they're negative or positive. Right, so here's a good example of polygenic. Right? You have one set of genes controlling whether you're A, B, or O, and then a second set of genes controlling whether you're positive or negative. Yeah, Jasmine? I mean, she pretty much answered it. Yeah, unless you know like a really good psychic. But otherwise, yeah, just gotta go and get some blood taken out. All right, so All right, height is another one. All right, so what are some factors that go into your height? Well, scholarship, but what dictates whether you're tall or short? What genes? Genes that control what? What about it? All right, so for one thing, growth rate, right? Your growth rate will be controlled by a gene. What else about your bones?
Density.
Perhaps how strong they are, right? You can't be too tall if your bones are brittle, right? So maybe your density. Do you think the shape of your bones has anything to do with your height? What if you've got little stubby leg bones?
Then you're really short.
All right, so then the shape of your bones, another gene is involved in your height. Right, so height is a polygenic trait. There's not just one thing and you're either tall or you're short. Right, multiple things are involved. Eye color is another one. So if I look at someone and they have brown eyes, is the brown the same the whole way through, from like the pupil to the edge of the iris?
No.
Has everyone noticed that there's like gradations, right? There's versions, right? So let's say, for example, if I look at these eyes, right? I did a little hard with the shade. Right, we've got a green eye here, a brown eye there, and a blue eye there. If you're blue-eyed, do all blue-eyed people have the exact same blue eyes?
No.
What are some things that might be different about the blue eye?
You can have like,
All right, it can be light or dark, sure. What else?
you can have,
All right, so we can have areas that are darker, like, right, it could have a rim, or it could be darker in the center. Right, so notice that, again, we have more than one gene controlling this here. Right, you might have a dark rim of blue on the outside, a lighter rim in the middle, or it might be darker in the middle, lighter on the outside. Do some folks have situations where their eyes might be a little bit of multiple things, like brown in some areas, green in others, right? Right, so once again, eye color is a factor of multiple things. Yay. All right, so now we're going to move on to some really good meaty adult high school stuff. All right, so next we're talking about how is it you figure out these kinds of things. All right, so who can tell me what this is from math class? Who knows what the FOIL method is?
First,
Function.
outer, inner, and then last.
Last, right? First, outer, inner, last, right? So folks have already learned this skill in mathematics. Now we use the same skill in genetics. So far we've been doing the little itsy bitsy baby middle school style Punnett squares, right? Where you track one trait, there's like 4 boxes. We're like totally over that and bored with it. Right, so we're moving on to the good high school stuff now. Right, so let's say we have multiple traits we're trying to track. Right, let's say we have a situation here where we're tracking not just one trait, whether you're big A or small a, but also a second thing. What do we call it if you have 2 different alleles, if you're like big and small at the same time? Heterozygous, right? All right, if I'm going to FOIL this, let's say this is someone's genotype, and I'm going to make a sperm or an egg, I'm going to make a gamete. What would the first thing I would write be, if I'm doing my first, outer, inner, last thing? So first I would have what? One big A, and the big A would connect to? The big G, right? Because that's the first thing of the second part, right? So we're thinking of it as being like this, right? If it's like algebra, right? So my first, right? So I've got my first here, my first there, right? Equaling that. What would be my next thing, right? So in my FOIL method, it's my outer, which would be what?
Big A.
The big A, you bet. And the little g, very nice. Right, so we have a big A and a little g. There's another sperm we've created, another gamete. All right, what's next? We have our inners,
the little A.
the little a and
Big G.
the big G. Awesome. All right, so we have little a, big G is my inner,
Little A, little G.
and then my last, my little a and my little g. Nice work, folks. All right, great. Let's talk about these gametes now, right? So here we have 2 dominant ones. one where we have a dominant and recessive together. Yesterday we had an expression for this, where this mixing happens without regard for what the other thing was. You don't rely on any other traits. Just like many of you don't get walked to the bus stop anymore. What do we call that? We're young adults that are on your own doing things. Independent, right? These things are independent. Right? Big A doesn't have to go with big G, and little a doesn't have to go with little g. Feel free to help her out. She might need a wet finger in her ear, or maybe even up her nose. But you can't stay here and not be conscious of working. So, awesome. All right. Right? All right, so we'll just make sure we got the right thing here, right? So we had those gametes, that worked out fine. Let's play with this. All right, so, folks notice there's rulers on their tables. You need a box like this one with 16 squares all together.
Can I put on this page, on the do not page?
Indeed. Right, we're going to wind up doing 2 of these. But this one will just be our skill practice one, and then our next one will have an actual scenario attached to it, like where we'll track a trait.
How many should the box have?
16 altogether.
How many cards?
4 and 4. All right, so,
What if you have other?
and what we're also going to do is we'll set our squares up, and then we're going to have some friends come up and foil our mom and dad genotypes for us.
Do we need to draw this?
Wait, are there?
Yeah. All right, so. All right, so again, making a box that has 16 squares, each square needs to be able to hold 4 letters. So just making it big enough that you can put 4 letters in each box. Whereas as before, we'd only do 2, right? So, because now we're creating these individuals that are going to have 2 of each allele. All right, and what we're moving on to now is what's called a dihybrid cross. Meaning 2, right? We're tracking 2 traits at the same time. And they can be totally independent of each other, right? You know, because just because you have blue eyes doesn't mean you have blonde hair. Just because you have curly hair doesn't mean the hair is black, right? These things are always in some random combination. Why don't we go ahead and come up with some scenario? What would we like A to represent? What trait would we like A to represent about an individual? And you can think about some of the traits we did in our experiment last week where we surveyed each other. We'd like to be eyes. All right. So what's big A going to equal? Or big A would have to equal brown eyes because blue would be the skip blue. All right. So big A equals brown eyes in this situation, and we'll have little a equal blue. Or do you want green? All right. We'll stick with blue. All right. And what about our G's? What would we like that to mean? And again, right, the letters are just things we make up as tools to track. Yeah, what would you like G to be? Any trait at all. Okay, it could be height, growth. All right, great. All right, so if you're a big G, you're going to grow up to be big and tall. And if you're a little G, you're going to stay small. All right, so, yeah, please do. All right, so what we're saying is that if you're an individual who has a big A, you have brown eyes, like our lady here. And if you have big G, you are tall, like this guy here. What does that mean for her? She's what? What are her 2 phenotypes?
She has brown eyes and she's short.
Brown eyes and short. Awesome. And what about this gentleman right here?
Brown eyes and tall.
Brown eyes and tall. Awesome. Super easy, right? All right, great. I'll do one, and then I'll invite some friends up. On our punnets, where do we usually put the male and female?
Male on top, female.
Awesome, right? So usually, right, we put the male up here on the x-axis, and we put the female down here on the y. All right, so let's say I'm going to foil mom, right? So I'll do my first 2, right? That one. So mom's egg, her first egg is going to be big A, little g. What will her next one be? All right, so I've done first It would be the same thing, Alvin, and why is that? Which G am I connecting to? The second one, right? So we'll do those two, right? This would also be big A, little g. Yeah, the intro should be a knot. And then it'll be the inner ones, right? Little a, little g for that A, and then the last A would be, right, this little a and that little g, right? So, cool. So what we did was we've made all of Mom's gambits, we made all of her A's. Now we need to say for Dad's sperm. Let's get some friends up here. Where's the cup here? There it is. All right, so if, Tiffany, if you could come up and do our F, our first. Gabrielle's not here. Ashley, if you could come up and do our O, our outer for Dad. Jasmine, if you could come up and do our inner, our I. And Jeremy, if you could come up and do our L, our last. It's always, there always has to be two, like, right, so. Very nice, good work. You are doing inner, so the third one. Awesome. Right, so you're foiling Dad, right, and you're doing the inner too. So is big A on the inside? I love watching you guys go through this whiteboard.
Put it down.
We only got two A's. We only got A's there. What? So
Oh, okay. I was looking at this.
Put it down.
Put it
No.
down.
Calm down.
I didn't even wait.
You're not up to that. Big A.
what kind of A you're gonna have?
Wait, geez.
Put it down. Yeah.
I'll throw it out.
So which two letters are on the inside?
Oh. Come on, Jasmine.
Little
Big G.
Jesus Christ.
All right, so takes a moment. So this one's on the inside,
Yeah.
and that one's on the inside. All right, cool. All right. Now that we've made our sperm and egg, what does each box represent? If we're combining a sperm and an egg, each box will be a zygote. All right, so let's get some friends. Wait, I'll do a zygote first, and then we'll get some friends up to do some other ones. All right, so I'm gonna use my traditional drag and drop method. So I'm gonna drag these two over. Right, so here's a big A. I'll drag over that little g. And then I'll drop that big A, and I'll drop this one. So what do we call this? If for eyes, we've got two big letters, that would be what? What do we call that? If you're dominant for both alleles. Homozygous dominant for eye color. So the eye color will be brown. And then here, if we have two, then the two alleles are the same but small. The person will be short, and we call it homozygous recessive. Nice teamwork. All right. It's not too obvious or anything. Just move from one bad activity to the next. I think you should come up and do the next box. Anyone you like. And obviously, like we're doing up here, you should be filling them in down there. All right, so you have to, we use a drag and drop method, meaning that these two here go in every one of those.
Oh my God.
And then those two go in every one of those. Choose anyone, pick any box you like and fill it in. All right, so again, feel free to go ahead and try it out. Try to fill these boxes in.
Like this?
All right, so right, you have that big A and that little G. Oh, yep. Very nice. Way to go. And let's see. Now I'll show you guys a second method some people like. It's not my personal choice, but some folks find it easier, right? So I actually do the drag and drop where I do all in one box at the same time. Some people like to do a more factory style method, where if I know I'm gonna be using this big A all the way across, they just go ahead and put that big A all the way across. They know that little G will go all the way across, so they put that little G all the way across. Right, you get the picture, right? And then they do the same thing going down. They say, oh, each of these will have a big A, so I can, and each of those will have a big G, so I'll bring that down. Either method is fine. You'll still, you'll wind up with the same steps. Oh wait, that's a little one. That's a little one. Right, so you should be doing just as I am, right? You should be filling in your dihybrid cross. So Eddie, I already know how to do this. It's not for me to practice, right? You're the one who's taking a test on Friday and an MCAS at the end of the year. Right, so, I did all right, but still. All right, and then a little G, and then I've got that one and that one. Right, so then either method's fine, dragging and dropping.
Shaquan Beckles, please come to the main office.
All right, now, the way this usually appears in the MCAS is they'll ask very particular questions about percentages. So we'll be practicing that in a moment. But for now, make sure you've got your chart filled out and, compare with mine. In an ideal world, we all have the same thing. Although, the universe is not always a perfect place of agreement and happiness. All right, and so. Okay, now what they'll do on the MCAS is they'll give you two parents, right? And they'll say, find out the parents' gametes, right? We did that step. We got their sperm and their egg. And then they'll ask you to cross the parents. We've done that, right? We've crossed them into these boxes. And then they'll ask you something like, what percent of the parents look like X, right? So let's try one of those out. So if I ask you, what percent of my parents, excellent. Oops, I'm sure it's okay.
Oh, yeah.
All right, so they might ask you something like, what percent of my zygotes are short and blue-eyed? So what do I have to look for? So in order to be short, I need what combination of A's? Or was it, I'm sorry, well, it was the G's for that, right?
Yeah.
You need two little G's, and if I want to be short, I need, oh, no, sorry, yeah. So I'm short, two little G's, and if I want to be blue-eyed, I need two little A's. So how many of my 16 fit that category? All right, so I've got
6,
5,
3.
whoa, okay. All right, so I've got those guys. Wait, did I do one of these wrong? Yeah.
The first row, the second column.
Oh yeah. All right. Thank you.
Oh, wait, that one? That one.
That one there. Yeah. Thank you. Wait, no, because that.
No, that one. Which one? Third.
The third row.
Tell me to go up or down.
Down.
Right. Other.
Yeah, that one.
It's a big G.
Oh, that should be a big G. Okay. Oh, gotcha. Okay, thanks. All right. So that means then only these two, right? Is that, we agree on that? Okay. So if I'm saying it's 2 out of 16, how do I convert that? What would that be? 2 out of 16. 1/8. Awesome. Right, and then you've got your answer, right? So 1/8, an eighth of my individuals have that, right? Let's say I changed it to tall and blue-eyed. I'll change the color there. So how many of these are tall and blue-eyed? What combinations do I need to be tall? All right, I could have capital G, or small and big. All right, so if you've got a big G anywhere, you're automatically tall. So we're looking for a big G with 2 small A's. Okay, 8. Right, so we're saying, right, 1, 2, who else? Which one was which again?
Big G.
Oh, any big G's, right? But we also have 2 small A's with our big G's. Is that one there? No. Until it's the end of the day, caffeine's worn out.
In the third column.
All right, so yeah, I've got this one, and that one,
That's it.
and that's it. Okay, so once again, so same kind of a thing, right? And you kind of get the picture there. So let's do it for a real scenario. All right, so let's have a look at this guy here. So this is the same person. This is a picture I got from the internet. And I did something to change his profile on either side. What's different about his face?
His legs.
What about that?
And that one, the third one's like, and like, it look like he got no upper lip.
What do we call this condition? Let's say this is a normal, symmetrical face, right? What would we call that situation? That would be an overbite. All right, so this person's got an overbite. And then obviously this guy's got a... Okay, now, we want to think about this from the evolutionary lens of mating. Because overbite and underbite are 2 sides of a coin, and one of them is considered more attractive sexually for a very particular reason of survival. So which side is more attractive? Is it these 2 or those 2? Is our overbite more attractive or is our underbite more attractive?
Right.
Alright, so how many folks think this guy is more attractive than this guy, right? How many folks would choose him over him? Alright, so we've got 3. How many folks that this one is more attractive than that one? 4. And how many folks find this one more attractive than that one? Alright, and that would be 7. Alright, now that is actually consistent with what we would believe. Let's say, for example, again, we're thinking about the evolutionary brain, why we adapt to certain things. Let's say I'm trying to be cute with somebody, right? And I'm making eyes at them, and I'm smiling. Do I go, or do I go, which one is cuter, having a little overbite or a little underbite? The overbite, right? We bite our all cute, like. Now let's think about this. Why would we choose people? Why would we choose mates that have an overbite? Let's say I'm holding a sweet, juicy, delicious apple, and I go to bite my apple. Which is a more efficient way of chewing? Or, right? Do we normally bite down on things or scoop them up? We bite down, right? So offspring that have a normal to an overbite survive better, right? They're more effective eaters, right? So our brain is designed to find mates that have this because they eat in a way that our species eats. All right, cool. So let's do a dihybrid for this situation, right? With overbite, we'll do this one and that'll be our class activity. Right, so overbite being the dominant feature, and once again thinking about independent assortment, right? Whether you're dark skinned and have an overbite, those things can come independently of each other. So let's do the same thing. We're going to FOIL for each parent, and then find our zygotes and males. Yeah. I know, 9th grade, we're still not sure.
May I get water after?
You have permission to get water afterwards.
Where's the gas? Just water.
Oh, that guy's long gone. I don't know where it's going. All right, cool. So we'll be doing the same thing with this, right? So we're going to, later on, by percentage, find out, well, how many percent of these have an overbite and dark skin? And how many percent of these have an underbite and light skin? So on and so forth. Right, so first thing to do, of course, is to FOIL to get your male gametes, to find your sperm. And then also FOIL the female, get